Today we will discuss a brief overview regarding how does the mechanism of titration work when it comes to determining the pI of an amino acid. In order to understand that, we need to know the principle of titration. What is titration? Titration is based upon knowing the concentration of your unknown solution. In this particular case, understanding the pKa of an amino acid. For this, what we require? One, a titrant, and second, an analyte. These are the two things that is essential. As a titrant, we should use a strong base over here NaOH of known concentration, and as an analyte we should use the amino acid solution. Therefore, before the starting point, before addition of NaOH, suppose an amino acid called glycine. We all know the structure of glycine, which is it is a protonated state. Now suppose NaOH is added drop by drop. What will happen? First of all, from this particular structure, we can clearly understand the CWH has a lower affinity, the O minus has a lower affinity towards the H plus. As a result, the alpha carboxyl group will start to deprotonate. The alpha carboxyl group will start to deprotonate. That means CWH will start to form CW minus plus H plus. Now, the fact is, when it comes to addition of NaOH, it's a strong base. It readily dissolves into Na plus OH minus. Now, most of my aspirants are asking, how come the release of H plus, whether it be from the carboxyl end or from amino end, is enhancing the mechanism, is enhancing the entire process of elevation of pH. As the number of H plus ions are increasing within the solution, they are supposed to lower the pH, but we are observing that the pH is being raised. How come? For this, I would like to make them clear about the fact that NaOH breaks down into Na plus and H plus. And whenever this NaOH is being used drop by drop into the amino acid solution, what is happening? The H plus from the COOH that is getting reprotonated, this H plus, it reacts with the OH minus from the NaOH, and hence water is formed. So therefore, there lies least amount of free H plus ion. As a result, the pH gets elevated. We all know that for alpha carboxyl group, the deprotonation starts from pH 2.2 approx, almost. And after the process is complete, after entire carboxyl group gets deprotonated, the deprotonation of alpha amino group starts near about pH 9.4. So therefore, the first deprotonation of alpha carboxyl, next second deprotonation of alpha amino. This one is near about 2.2, this one is near about 9.4. So therefore, the pi of the solution should be 2.2 plus 9.4 divided by 2, which is around near about 5 point something approx. So therefore, near about this pH, 50% of the amino acids, 50% of the amino acids gets deprotonated. This is considered as the Zwitter ionic state. For glycine, it will be like COO minus NH3 plus H and H. As simple as that. Now, interesting fact is this is something without any side chain. What happens when our amino acid with side chain? The mechanism is quite different. If the side chain is suppose a carboxyl group, then what is the mechanism? Suppose at pH previously you have known 2.2 around, alpha carboxyl gets deprotonated or starts to deprotonate. Now next comes the deprotonation of side chain carboxyl group, which is around suppose 4.1 at pH. So therefore, and for alpha amino, we all know this is 9.2 or something like that pH. It starts deprotonation. So first deprotonation will take place from alpha carboxyl, second deprotonation from side chain carboxyl, and third deprotonation from alpha amino group. So from this, if we need to calculate the pi, we have to understand that after the start of deprotonation from alpha carboxyl group, the net charge will still be plus 1. And when it comes to deprotonation from alpha carboxyl group, side chain carboxyl group, then the pH of this entire amino acid will reach to 0. That means typical neutral state. So therefore, the Zwitter ionic state will occur from or rather in between alpha carboxyl to side chain carboxyl. Therefore, pi for that particular amino acid containing side chain carboxyl group should be 2.2 or 2.1 plus 4.1 divided by 2, which comes to 3.1. This is the state where it will become zwitterionic. This lies for alpha carboxyl. What will happen if our side chain is a amino group? So first deprotonation as usual from alpha carboxyl group near about pH 2.2. Second deprotonation. Now, as there is no carboxyl group left, the second deprotonation will definitely start from alpha amino group as there is no side chain carboxyl group, which is near about at pH 9.4, suppose, near about. And the third deprotonation will start from side chain amino group, which is near about 10 approx. Therefore, you can see that the Zwitter ionic state, if we release the H plus group from the alpha carboxyl group, the charge will still be plus 1. So the net charge 0 will starting from or will the net Zwitter ionic state will be achieved in between the deprotonation from alpha amino group up to the deprotonation of side chain amino group. These two facts. Therefore, in this case, where the side chain of an amino acid contains amino group, in that case, the pi will be pK2 plus pKa3 by 2. That means 9.4 plus 10 by 2 near about 9.75 approx. So this is how the entire process of calculation is being done when it comes to pi. And not only that, this is the mechanism where the H plus, will be no H plus within the solution as the strong base is absorbing the H plus and giving rise to H2O. Hence, there will be no free H plus within the solution, and this will finally lead to the rise in pH. I hope you have understood the entire thing for this video.